Welcome to this explainer. We're jumping right into a microscopic world that is quite literally sitting on the tip of your nose right now. Today, we're unpacking the absolutely fascinating dual nature of a very specific bacteria. It's basically a classic tale of Dr. Jekyll and Mr. Hyde, and it's playing out every single second on your skin. So let's dive into it. Okay, this might sound like straight-up science fiction, but it is absolute medical fact. There is a bacteria thriving on you right now as you listen to this. That, under the wrong circumstances, could severely complicate your next surgery. We're talking about an organism called Staphylococcus Epidermidis, and understanding its double life is absolutely crucial for modern medicine. Section 1. The bug on your skin. So Staphylococcus epidermidis is, believe it or not, a totally normal resident of the human body. It abundantly colonizes our skin epithelium, our mucous membranes, and even the external auditory canal right inside your ears. Now, in healthy individuals, it's ubiquitous, entirely non-pathogenic, and it actually acts as a sort of microbial shield. By taking up prime real estate on our skin, it literally helps protect us from much more dangerous foreign invaders. Now, if you're looking at this from a lab perspective, what really sets this bug apart is that it's famously coagulase negative. Because it doesn't produce the enzyme coagulase, lab technicians immediately know they're dealing with a milder, typically less aggressive strain compared to the other major pathogens out there. It's a massive cheat sheet distinction that immediately directs the initial medical approach. To really get a feel for this, let's stack our microscopic neighbor up against its much more famous and aggressive cousin, Staphylococcus aureus. While S. aureus is coagulase positive and causes rapid skin infections and abscesses, S. epidermidis is coagulase negative and highly sensitive to an antibiotic called novobiocin. Instead of causing those aggressive skin abscesses, it is the undisputed master of the strong biofilm, and it specifically targets prosthetic devices. Section 2. When good bacteria break bad. So when exactly does this friendly neighbor turn into a nightmare? Well, S. epidermidis is an opportunistic pathogen. It exploits human vulnerability. The exact moment this friendly flora breaks bad is when a foreign biomedical implant is introduced into the human body. Something like a catheter entering the heart provides the absolute perfect, pristine, abiotic surface for the bacteria to bypass our skin barrier, colonize, and take hold. Basically, if we're putting hardware inside the body, it's at risk. We're talking prosthetic heart valves, IV and central venous catheters, artificial knees and hips, CSF shunts in the brain, and even pacemakers. And the terrifying thing is, these infections are incredibly sneaky. They are often indolent, meaning they grow super slowly without obvious systemic signs of inflammation right off the bat. A patient might just have an intermittent fever or maybe some subtle diffuse pain around the implant. Section 3. Building the biofilm fortress. Which brings us to its ultimate superpower, the primary virulence factor. Once S. epidermidis actually attaches to a knee replacement or a pacemaker, why is it so ridiculously hard to get rid of? Well, unlike other bacteria that secrete aggressive toxins to destroy human tissue, this guy uses surface-anchored proteins to bind to our own extracellular matrix proteins. Then, it secretes this incredibly thick extracellular slime layer, an exopolymer matrix. It is quite literally building a wall around itself and its community. I mean, it's basically a microscopic force field. But here's the crucial part. This self-produced slime isn't just physical armor. It fundamentally changes how the bacteria live. Deep inside this biofilm, the bacteria actually downregulate their metabolism. They go into a sort of hibernation. And because most of our antibiotics target actively dividing high metabolism bacteria, the drugs just wash right over the biofilm without killing the dormant bugs inside. It completely shields them from our white blood cells and leads to severe antibiotic treatment failure. Section 4. The multi-drug resistant threat. 75 to 90%. Let that sink in for a second. That is the staggering percentage of healthcare-associated strep epidermidis strains globally that are now resistant to methicillin. Because this bacteria is just hanging out as a normal resident in hospitals, on the skin of patients and healthcare workers, it has had decades to horizontally swap genes with other bacteria and build up some incredibly robust resistance. And things are definitely escalating. A recent rapid risk assessment by the European Center for Disease Prevention and Control highlighted a pretty chilling reality. They found a previously unrecognized international spread of near pan-drug-resistant strains. We're looking at genetic lineages known as ST2 and ST23 spreading globally across healthcare systems.
It is making infections of indwelling devices incredibly terrifying to treat.
Our antibiotic arsenal is shrinking, and it's shrinking fast. Methicillin? Mostly useless now. Rifampicin is often recommended to penetrate those biofilms we talked about, but we're seeing dual gene mutations neutralizing it, meaning it can absolutely never be used alone. Vancomycin is traditionally our heavy hitter, but strains are developing heteroresistance. These are subpopulations that survive the drug completely undetected by standard tests. And linezolid? Well, resistance to that is now spreading rapidly via mobile genetic plasmids.
Section 5. Stopping the superbug.
So how on earth does modern medicine fight back against a heavily armored, drug-resistant bug that literally lives on our skin? First and foremost, prevention is everything. Strict skin disinfection before surgery is absolutely paramount. Second, because of the wild drug resistance we just looked at, doctors have to run individual antimicrobial susceptibility testing for every single patient's specific strain. But the third step is by far the most sobering. Because antibiotics usually can't penetrate that biofilm force field, just medicating the patient isn't enough. In the vast majority of cases, surgeons literally have to go back in and surgically remove or replace the contaminated hardware.
If you take absolutely nothing else away from this explainer, remember this core profile. S. epidermidis equals normal skin flora, it's coagulase negative, it's novobiosin sensitive, it relies heavily on building a biofilm to survive, and it primarily causes prosthetic device infections. That one single profile completely covers the crucial duality of this bug, capturing exactly why it's both a friendly shield and a truly formidable foe. I want to leave you with something to really chew on. We have vaccines for so many deadly diseases, right? But why is it so ridiculously difficult to develop a vaccine against S. epidermidis? Think about it for a second. This bacteria protects us daily from much worse invaders. If we just wipe it out, something else is going to step in and take its place. So how do you train the human immune system to attack a bug only when it crosses a boundary, without destroying the delicate balance of your normal human microbiome? It is a massive, incredibly complex challenge for the future of microbiology. Thank you so much for joining me on this explainer. Keep asking those big questions, and I'll see you next time.